Okay, so today we're talking about the urinary system, and another name for the urinary system is the renal system. So we'll be taking a close look at this system. We have different structures in the system. The kidneys are the main structure, of course, in this system. There's two kidneys and they produce urine, and there's a urinary tract then. From the kidney is a ureter from each kidney. So there's a right and a left kidney, and the ureters extend from the kidneys, and then the ureters will drain into the urinary bladder, right? So there we go. And then from the urinary bladder, there will be one urethra, okay? And so the tract from there is a little bit different between the male and the female, but this is our urinary tract. Now, when we look inside the bladder then, the urinary bladder, where the opening is going up into the ureters, we see little openings there, and then there's a little opening going into the urethra. And if we connect those dots together, it looks like a little triangle, and we call that little triangle the trigone. Okay, so that I think is one of the structures that you have on your lab list. Okay, so what are some functions of the urinary system? So first of all, the kidneys are such a huge important part of homeostasis in your body. They function to regulate blood volume. So they're going to keep your blood volume where it's supposed to be, and since they're keeping the blood volume where it's supposed to be, it also regulates blood pressure, because blood volume has a lot to do with blood pressure. Second of all, it regulates plasma concentration of ions, so ion concentrations. It will determine what your plasma concentrations should be for sodium, potassium, chloride, calcium, and it keeps those in the right concentrations that they should be in your blood. 3, it helps to stabilize pH of your blood. Okay. And then 4, it helps you to conserve nutrients so that you're not getting rid of nutrients. You're keeping nutrients like glucose and amino acids so that you can make ATP. And at the same time, it removes wastes. So it's keeping the good stuff and it's getting rid of the bad stuff. And then lastly, it detoxifies poisons. So in this, and toxins. So in this case, it kind of helps the liver because the liver is doing the same thing. And in addition to detoxifying these toxins and these poisons, it'll also deaminate amino acids during starvation. Okay, so when that happens, of course, then we have a toxin that's called ammonium that's produced, and ammonium has to be converted to urea, which can then be urinated out. Right, a couple other things about the anatomy. So the left kidney would be higher than the right kidney. I don't have the right kidney drawn in here, but the left kidney is more superior. And each of the kidneys are capped by a gland that we call the adrenal gland. So the adrenal gland is our stress gland. There is an adrenal medulla, which is right here. And then there's an adrenal cortex, which is right here. One of the hormones released from the cortex is called aldosterone. And aldosterone will target the kidney and cause the kidney to reabsorb sodium, and then water gets reabsorbed as well. So we'll be going into that. Now, some other things about the kidney. The kidneys are what we call retroperitoneal. Retroperitoneal. Now, if you remember what the peritoneum is, there's the visceral peritoneum that covers each of the individual organs, and then there's the parietal peritoneum that lines the peritoneal cavity. Well, these kidneys sit behind that peritoneum, so they're not in the peritoneal cavity. So what that means is that when a person has a kidney infection, for example, they will probably have more of back pain than they will of abdominal pain. If the organ was inside the peritoneum, then they'd have more abdominal pain or abdominal pelvic pain, but because the kidney's behind the peritoneum, it's in between the peritoneum and the spine, they're going to end up with back pain. And so how can we distinguish this in patients? Well, not only will they have back pain if they have a kidney infection, they'll also have a fever. So that's one thing to look out for. And there's some other organs that are retroperitoneal, and you need to memorize what those organs are because all of these organs could potentially lead to back pain. And so we use the acronym SAD-Pucker. So we use the initial letter, and so the S stands for suprarenal. So suprarenal gland is actually the adrenal glands. The A stands for the aorta and also the inferior vena cava. So those are also retroperitoneal. The D stands for duodenum. That is the initial portion of the small intestine. The P stands for pancreas. U for ureters. C for colon. K for kidneys. E for esophagus, and R for rectum. So the kidneys have an outer capsule, right? Most organs are going to have an outer capsule, and so this outer capsule we call the fibrous capsule of the kidney, right? And then on the inside of the kidney, we have two layers. We have the cortex on the outside, and then we'll have the medulla on the inside. And then the very outside, that hard covering, is called the renal capsule or the fibrous capsule. Then outside the capsule, there's going to be a layer of fat that we call the perinephric fat. And that kind of cushions the kidney. And then outside of that, there is a layer of renal fascia. So there's going to be a layer of renal fascia out there, and then the renal fascia is going to connect that kidney to the surrounding areas. Where the ureter exits from the kidney, that area is called the hilum. And the other things that are going to enter and exit through the hilum are the renal artery, and then we have the renal vein. So those things are the three things going in and out of that hilum area. So again, we've got the ureter, and we have the renal artery, and we have the renal vein. The kidney is about 4 inches long, and it's going to be about 2.5 inches or 2.2 inches wide, and it'll be about 1.2 inch thick. Okay. It weighs about 5.25 ounces. Now let's take a look at the inside or the internal anatomy of the kidney. All right, so here we have the kidney like that. Here's the ureter coming out like this. And right inside here, we have an expanded area that we call the pelvis. That's the pelvis of the kidney. And the pelvis is going to receive urine from these other areas that are called the major calyces. So those areas there are major, that's a major calyx. Okay, and so plural it would be calyces, if we were to look at it plural. And then each one of those major calyces, we have these other little tubes that are draining into there. And there could be 2, there could be 3, but the smaller ones there are called the minor calyces, or minor calyx for single. So when urine is being produced, it drains into the minor calyx, then into the major calyx, then into the renal pelvis, and then into the ureter. And from there, the urine will drain down to the urinary bladder, where it's stored until you urinate it out. At the tip of the minor calyces is a structure called the renal pyramid. Right? So each one of these minor calyces has a renal pyramid right at the beginning of it. So I can draw all of them in, I guess. Like that. So all of these pyramids. And so the pyramids are part of what we call the renal medulla. So this whole area here, that's the renal medulla, where all of those renal pyramids are found. And in between the renal pyramids, so that's a renal pyramid, in between the renal pyramids we have these columns. So those are just renal columns. So on the inside here, this whole area is the medulla. And then anything outside that is called the cortex. In a little bit, we're going to be looking at the functional structures of the kidney, which are called the nephrons. And the nephrons start in the cortex, and then they extend down into the renal pyramids, into the medulla. So we'll be looking at those. They're out here, and they are these tubules, and they extend down. Okay, so we'll be looking at those in another picture. Next, though, I just want to show you how the renal artery is going to bring blood into the kidneys, and the renal artery, as it branches off, has to bring blood into those little nephrons, and that's how that blood is going to get filtered. So what we have to show you now is how does the blood get from this renal artery here all the way out to this little capillary bed, which is called the glomerulus. So that's what we're going to look at in the next slide. So I'm just going to draw here one of the pyramids. Okay, so this is obviously the renal medulla. And then out here is the cortex. And then out here in the cortex is where we have that thing that we call the renal tubule. Right, so it's going to reach down. All right, we'll go over that nephron later. But we have to start by looking at the renal artery. So the renal artery is entering into the kidney. This is the renal artery. And it's going into the kidney. Right? So the renal artery, first of all, is going to divide, and it's going to become the segmental artery. Right? Then the segmental artery is going to divide, and it's going to follow up in between two different pyramids. So when it's going up in between two different pyramids like that, we call that the interlobar artery. Then the interlobar artery is going to split off and it's going to arc around the pyramids. So as it's arcing around the pyramid on each side, we will call that the arcuate artery. From there will be little cortical radiate arteries that come off of that arcuate artery. So they're smaller little arteries, and they're radiating off of that arcuate artery. So we call those the cortical radiate arteries. From the cortical radiate artery, we have an artery that goes into the nephron, and that is actually an arteriole, and we call that the afferent arteriole. All right, so now we're getting a lot smaller. Remember, arteries become arterioles, arterioles then branch into capillaries. So this afferent arteriole is going to branch off into a capillary bed, but it's kind of a special capillary bed because normally capillary beds then will become venules. But in this case, this is just an expansion of that afferent arteriole, and then coming out of that afferent arteriole, we have the efferent arteriole. So we still are in the artery, the group of arteries. It's not yet a vein, right? So that's the efferent arteriole. And the little cluster of capillaries that we have there, that's called the glomerulus, right? So now what's going to happen to that efferent arteriole? The efferent arteriole then will branch off to become capillaries, and those capillaries we call the peritubular capillaries. And we call it that because they are surrounding that tubule of the nephron. And they'll surround the entire nephron loop, and they'll surround the entire proximal and distal convoluted tubules. And so they're surrounding that tubule, and then those peritubular capillaries are going to converge back together, and they will become, as they converge, they are dropping off oxygen at that point in those peritubular capillaries, and they will converge to become the cortical radiate veins. So now we've got all these cortical radiate veins. Those cortical radiate veins then drain into the arcuate vein. The arcuate vein then drains into the interlobar vein, and the interlobar veins then drain directly into the renal vein. All right, so a big difference here, we have blood going in, and then it's kind of coming out the exact same pathway. The big difference here is that there is no segmental vein. All right, so let's work together here and follow the pathway of the blood all the way into the nephron and then all the way out of the nephron on this figure. So the first thing we're going to look at are the arteries. And so we're going to start here with number 8, and this is the interlobar artery. Right, so we already came in from the renal artery to the segmental artery, and now we are seeing the interlobar artery. The interlobar artery then will divide and will go over the top of those renal pyramids, and so number 6 then is the arcuate, as they arc around the top of those pyramids, the arcuate artery. The arcuate artery then has these radiating arteries coming off of it, which are called the cortical radiate arteries. And then the cortical radiate artery will branch, and that branch there is number 2, and that is called the afferent arteriole. The afferent arteriole then branches into these capillaries, this group of capillaries inside the nephron here. We can see these branches, these capillaries, and that is called the glomerulus. So number 15 is the glomerulus. From there, the glomerulus will drain into another arteriole. So this is still part of the artery system, and so that's number 3, and that is called the efferent arteriole. And the efferent arteriole then is bringing blood around here, and we can see that it branches off into these little capillary beds, and these capillaries wrap themselves around the tubule, around the nephron tubule. And so that those capillaries are called peritubular capillaries. So as those peritubular capillaries wrap themselves around all those pieces, all those tubules of the nephron, they're distributing oxygen, and this is where nutrients are going to be reabsorbed. This is where things will be secreted and reabsorbed. At the end, though, as those little peritubular capillaries are dropping off oxygen, then those capillaries are going to merge back together to become venules, right? And then they're going to become veins. So now they look more blue because they've dropped off their oxygen and they have become little venules, and those little venules will then start converging into veins. So now we see the first vein as those peritubular capillaries are converging together, and that vein is called the cortical radiate vein. The cortical radiate vein then drains into the arcuate vein. The arcuate vein then drains into the interlobar vein. And then there is no segmental vein, so then the interlobar vein drains into the renal vein. So now that we know all of the blood as it's coming into the nephron area and then exiting, so now what we're going to look at is the actual nephron itself. So the nephron is a tubule, that's all it is, and the tubule has different sections to it. So now I want you to take a look at the different sections. So the first section I want you to look at is right where that afferent arteriole comes into the nephron, and that part right there is called the glomerular capsule. So inside the glomerular capsule, we have the glomerulus, which is that little group of capillaries. So together, the glomerular capsule and the glomerulus, we could actually term that the renal corpuscle. It contains both of those. Then the next section that we see of the nephron is leading right away from that glomerular capsule, and that is called the proximal convoluted tubule. So, in other words, the PCT, proximal convoluted tubule. It's convoluted because it has lots of turns in it. It is proximal because it's closest to the that glomerular capsule. From there, the proximal convoluted tubule will lead into the nephron loop. The nephron loop has a descending limb, and this is as fluid is flowing down. That's why we call it descending. And then it has an ascending limb. And that's because the fluid will be moving upward or ascending. The other thing that we can see is that portions of the descending limb are thick and portions are thin. Portions of the ascending limb are thin and portions are thick, so we give them separate terms as well. Mostly what we'll be talking about is the descending thin limb, or the DTL, which is thin, and the ascending thick limb, or the thick ascending limb, which is right there. So we'll be talking about the DTL and the TAL as we go through here. And I'll be sure to actually not just use the abbreviations, I will use the actual terms. From the thick ascending limb of the nephron loop, the tubule becomes the distal convoluted tubule, right? So it also is convoluted, and we can call this the distal convoluted tubule. It's distal because it's furthest away from the glomerular capsule. So that's why we call it the distal convoluted tubule. And then the distal convoluted tubule is going to lead into a collecting duct. So here we have the collecting duct, and there are many different distal convoluted tubules that will be draining into the collecting duct. The collecting duct then will drain into the renal papilla, and the renal papilla will then drain into the minor calyx. The minor calyx drains into the major calyx, then into the renal pelvis, and then finally to the ureter. So what we're going to be doing throughout the rest of this chapter is we're going to be looking at each one of these separate segments of this nephron, and we're going to look at what is the function of each one of these separate areas. And then we're going to look at the formation of urine, because by the time that fluid moving through the tubule gets to the collecting duct, we now call it urine. So while that fluid is in all of these proximal convoluted tubule and the nephron loop and the distal convoluted tubule, all along the way there, we refer to it as tubular fluid, right? And the space surrounding the tubule we call the peritubular space, or the peritubular tissue, or even the interstitial tissue. And then you have the blood vessel on top of that. So, but in the tubule, we move from tubular fluid, and then finally in the collecting duct, it becomes urine. We call it urine then. So let's look quickly at the portions of the nephron again. We have the capsule there, which is called the glomerular capsule. Inside the glomerular capsule is the glomerulus. From the glomerular capsule, we have the proximal convoluted tubule. The proximal convoluted tubule leads into the nephron loop, which is this whole thing. The thin portion of the nephron loop, as it's descending, is called the descending thin limb. The descending thin limb loops around to become the ascending thin limb, and then we see a widening here of the tubule, and that is called the thick ascending limb. Now, one thing to note is, even though the descending limb, this thin limb here, and the ascending thin limb, they look like they have a more narrow lumen, they actually don't. It's just the thickness of the cells that line those tubules. So up in the thick ascending limb, those are going to be more cuboidal cells. So it makes that thick ascending limb appear thicker. And in the thin limb, it's more squamous cells, which are thinner, and that's what makes that tubule appear thinner in those areas. But the lumen itself is going to be just the same in the thick limb as it is in the thin limb. The nephron loop leads into the distal convoluted tubule, and the distal convoluted tubule drains into the collecting duct. All right, now one thing I want to note right here, though, is that we see this nephron as it's all spread out, and that's not actually what happens. It kind of bends over on itself so that there's cells, portions of the distal convoluted tubule. So I'll be drawing in some little cells here that are lining that distal convoluted tubule. Because it's bent over, those cells in the distal convoluted tubule will actually be over here, coming in contact with this afferent arteriole and with cells of the afferent arteriole. Where the cells of the distal convoluted tubule come in contact with the cells that are surrounding that afferent arteriole, we give that a special name because that has a special purpose. So where those two cells, where those groups of cells come together, we call that the juxtaglomerular complex. Juxtaglomerular, meaning juxta is next to the glomerulus, complex. Those special cells act as either baroreceptors or chemoreceptors. So let's take just a little closer look at that juxtaglomerular complex. So right here, I'm going to draw the afferent arteriole. Right, and that's going to go into the glomerulus. And then the nephron loops back around so that the distal convoluted tubule kind of goes right underneath that afferent arteriole, like that, right? And so the cells lining the distal convoluted tubule here are going to come in contact with cells that surround the afferent arteriole. Right? So the cells are called the macula densa cells if they are part of the distal convoluted tubule. And then of the cells that are surrounding the afferent arteriole, we call those the juxtaglomerular cells. The juxtaglomerular cells are actually modified smooth muscles of the arteriole, of that afferent arteriole, and they're the ones that act as baroreceptors and chemoreceptors. So they're detecting blood volume in that afferent arteriole, and they're also detecting chemicals like oxygen and as well as calcium and hydrogen. So they're detecting these things in the afferent arteriole. The macula densa cells, on the other hand, they are going to be monitoring what's going on in the distal convoluted tubule. So they are they're trying to detect the osmotic pressure in the distal convoluted tubule, meaning they're looking at the solute concentration and trying to determine if the fluid that's in the distal convoluted tubule, which is going to quickly become urine, how concentrated it is. Okay, now another thing that those juxtaglomerular cells do, though, besides monitoring what's going on in that renal artery or what's going on in that afferent arteriole, is that they will secrete renin. So those cells secrete renin or renin, however you want to say it. And this is a very important hormone because when that gets secreted, it's going to increase your thirst because renin goes to the hypothalamus and stimulates your thirst center. It also causes the release of ADH from the posterior pituitary, and it also causes the secretion of aldosterone from the adrenal cortex. So before we start talking about the functions of the nephrons and each of the individual locations of the nephrons, I do want to go over the different types of nephrons. So there are two different types of nephrons, and the first type is called the cortical nephron. So the cortical nephron has a shorter nephron loop. And so since it has a shorter loop, it's not going to be going into the pyramid as much. So here's the pyramid, and then here's that cortical nephron. There's your proximal convoluted tubule. There's your nephron loop. Okay, so what we see is that it doesn't go as far into that renal pyramid, so it's not going as far into the medulla. So it has a short loop. 85% of nephrons are these cortical nephrons. With this type of nephron, the efferent arteriole becomes the peritubular capillaries, and then the peritubular capillaries become the cortical radiate vein, and then we lead out of the kidney the way that I had showed you before. The second type of nephron, though, is called the juxtamedullary nephron. Juxtamedullary nephron. And so the juxtamedullary nephron has a long nephron loop. And 15% of all nephrons are the juxtamedullary nephrons. So if we draw those out, here's our pyramid. Here's our juxtamedullary nephron, long loop. Okay, there's the collecting duct. All right, so what we see here is that we have a much longer loop. We also see that the nephron is closer to the medulla, right? So the medulla starts where that pyramid is. That's why we call it juxtamedullary, next to the medulla. Whereas the other one is cortical nephron because it's farther away from the medulla and it's more in the cortex. With the efferent arteriole of the juxtamedullary nephron, the capillaries, the peritubular capillaries, have to wrap themselves all the way down this really long loop here, and then they will become the cortical radiate veins and lead their way out of the kidney. So we give a special term to this really long peritubular capillary, and it is called the vasa recta. So the vasa recta are found then in these juxtamedullary nephrons. So because this is so long, since that nephron loop is so long, there is a lot longer time where urine can get concentrated. So in this nephron loop, we're going to see that sodium get reabsorbed, sodium and chloride ions get reabsorbed, and that causes water to get reabsorbed. And so because this loop is so much longer, and we are reabsorbing water there, the urine then becomes more concentrated. And so it's these types of nephrons, these juxtamedullary nephrons, they are the ones that enable the kidneys to produce concentrated urine. So next let's talk about some metabolic wastes. So one of the things that the kidneys do is they help the body get rid of metabolic wastes. So the metabolic wastes end up in the urine, and you get rid of them, get them out of your body. Our bodies form 3 important metabolic wastes. The first one is called urea, and we've talked about this before. Urea is the most abundant metabolic waste, and it's from the breakdown of amino acids. So if you remember, when amino acids get broken down, then they form this toxic ammonium, and then ammonium has to be converted to urea during the urea cycle, and then the kidney can get rid of the urea. The second metabolic waste that's produced is called creatinine. Creatinine is produced when skeletal muscles break down creatine phosphate. Remember what creatine phosphate is. It helps to store ATP. It stores that last phosphate group for ATP until ATP needs it back. The last metabolic waste in the body that I want to mention is called uric acid. So uric acid is from the breakdown of RNA. It's really from the breakdown of those nitrogenous bases in RNA. So you remember what those are. Those are uracil, adenine, guanine, and cytosine. So all of these metabolic wastes have to be dissolved in water in order for them to be excreted. And since they have to be dissolved in water, getting rid of waste products involves water loss. So that is just an inevitable type of water loss. There are 3 distinct processes that occur in the nephron tubule, and I want to go through those first so that you understand what I'm talking about when I mention them. So the first thing is filtration. Filtration is when blood pressures force water and solutes through a membrane. So you have this filtration membrane, and you have blood pressure pushing fluid up against that membrane. And what comes out on the other side of the membrane is water and ions and small molecules. Right? So the majority of filtration occurs because blood pressure is pushing blood up against this filtration membrane, forcing water, ions, and molecules across, but keeping things like red blood cells and white blood cells and little plasma proteins in the blood. Reabsorption is the next process that I want to mention. And reabsorption is the removal of water and solutes from the tubule into the peritubular space, or the peritubular fluid, and which is also the interstitial fluid. That is the fluid surrounding the nephron. And then eventually it will be moved into the peritubular capillaries. So now it's back into the body. So it's moving from the tubule back into the body. Then the third type of process is called secretion. And secretion is the transport of solutes from the peritubular capillaries into the peritubular fluid, which then moves into the tubule. Let's just take a quick look at this. I'm going to draw, here's the renal capsule, there's the proximal convoluted tubule. I'm just going to draw part of this proximal convoluted tubule. Then we're going to look at the blood coming into the renal capsule and then into the glomerulus. So there's a bunch of capillaries in there, and then eventually out into that efferent arteriole, right? And then the efferent arteriole becomes the peritubular capillary. So let's just draw this out and let's name a couple of things here. So this is the afferent arteriole. This is the glomerulus. This is the efferent arteriole. And this is the peritubular capillary. Then we can continue naming some things. You know what I'm talking about. This is tubular fluid. This is the peritubular fluid. This then inside the peritubular capillary is the plasma. And then we also have filtrate, which would be the fluid that's in between the glomerulus and that capsule. That is the glomerular capsule. So let's take a look at these processes again. So the first process that I mentioned was filtration. Filtration is when the blood pressure is pushing up against the walls of the glomerulus and it's pushing through fluid. So fluid, we have water that comes out and is in the filtrate, and we have ions and nutrients and wastes, of course. So we've got all of those things in the filtrate, and then the filtrate moves into the tubule. And as soon as it moves into the tubule, it then becomes called the tubular fluid. All right, so this process that occurs right here, you know, right there where the blood pressure is pushing up against that glomerular lining of the capillary and pushing those things through, that is what we would call filtration. And that is the only place where filtration occurs, and we're going to take a really close look at filtration. Then we have that process which is called reabsorption. So reabsorption is when these things that are in the tubular fluid, those things like waste products, and not waste products, but things like water and ions and nutrients, those are things that we really don't want to lose. We don't want them to end up in the urine. So we want to reabsorb those things. So those things are going to move from the tubular fluid into the peritubular fluid and then into the plasma. That's what's called reabsorption. Now we have it back in the blood, and we've been able to keep these really important things like water, ions, nutrients. These are things that are going to be reabsorbed. Now, what we're going to learn is that some of these things are going to be reabsorbed through simple diffusion. And you remember what that is? That's just moving solutes from an area of high solute concentration to an area of low solute concentration. Sometimes this reabsorption might use carrier proteins. So when carrier proteins are used, sometimes it's co-transport, where they're transported across with another ion. Sometimes it is counter-transport, where they're transported in the opposite direction of ions. And then the substances will eventually lead into the blood, so they re-enter the blood. Water is going to be reabsorbed through osmosis. But these other things, ions and nutrients, will be reabsorbed through simple diffusion or carrier proteins. Secretion then is when solutes from the peritubular fluid here, which actually came from the plasma, so we have these solutes or waste products. They are coming from the plasma into the peritubular fluid and then into the tubular fluid. That is called secretion. Secretion is necessary because sometimes filtration doesn't get all of the waste products. And so then as the plasma travels through those peritubular capillaries, this secretion gives us yet one more chance to get rid of those waste products. It also helps us to get rid of things like ions like hydrogen if the blood is too acidic. Because remember, if the blood is too acidic, it means it has too many hydrogen ions, and so we can get rid of those hydrogen ions during secretion in this tubule. So it's basically secretion will lower the plasma concentration of undesirable materials. It basically provides a backup to filtration. Secretion ends up being the primary method of excreting drugs. The drugs are often missed during filtration, and so then they have to be eliminated through secretion into the tubule. So normal kidney physiology is going to continue as long as filtration, which is over here, and reabsorption, which occurs right here, and secretion, which is occurring in this direction, as long as they are functioning properly. All of these processes have to function in very narrow limits. A disruption in kidney function has an immediate effect on the composition of the blood. So it's important that all of these things, filtration and reabsorption and secretion, are occurring normally. And if they're not, then it's going to change the composition of the blood significantly. If both kidneys are disrupted, then death could occur within days. So what we're going to do now is we're going to go through each of the different processes, and I'm going to start with filtration. And then we're going to stop for this video, and then I'll create another video to look at what happens in the proximal distal convoluted tubules and the nephron loop, as well as the collecting duct. But we're going to start here with the process of filtration. And what I told you before was that filtration is due to hydrostatic pressure from the heart beating, and it causes fluid and ions and small molecules to be filtered across the glomerular capillaries. So when we're talking about filtration in the glomerular capillaries, we call it glomerular filtration. So we've seen filtration before when we were talking about just regular capillaries in the body. But now we're going to talk about glomerular filtration. It's a little bit different because there are some other structures involved that we don't see in our other body capillaries. So filtration here in the glomerulus is the first vital step so that waste can be excreted, and so pH can be controlled and blood volume can be controlled. It works kind of like a drip coffee machine. So even though there's hydrostatic pressure pushing up against a membrane, what comes out on the other side is more like a drip. Okay, just like a coffee filtration machine. So filtration involves this thing that we call the filtration membrane. And the filtration membrane is made up of three things, basically. We have the endothelium from capillaries, we have a basement membrane, and then we have these other cells surrounding the basement membrane, which are called podocytes. And the foot processes of those podocytes contain little filtration slits. So we're going to take a look at these three layers of this filtration membrane. And we'll just kind of draw it out here. So we'll see that we have, first of all, the basement membrane. So let's just draw in the basement membrane here. So this is a basement membrane. And on one side of the basement membrane, then, we have these blood vessels, the glomerular capillaries. And the walls of the glomerular capillaries they're made up of these endothelial cells. Okay, so we have, these are the endothelial cells of the capillaries, and then this is the basement membrane, And then on the other side of the basement membrane, we have these podocytes and their foot processes. So there's this big podocyte, and then they have these foot processes here, right? These are its foot processes. So this is a podocyte. There's its nucleus. And these are its feet. And then this is a filtration slit. So when the blood is pushing through, the heart beats and blood is coming in through that glomerulus, and it hits the walls here, it has these three things that it has to make its way through. It has to get through the endothelial cells, or it has to get in between the endothelial cells. Then it has to make its way through the basement membrane. And then it has to make its way through those filtration slits of the podocytes. It's those three things that allow the passage of water and solutes and ions. So let's start by looking at the endothelial cells. First of all, we know that inside the blood there are these big red blood cells. The red blood cells are too large to get through even the endothelial cells, so the red blood cells will take a pass and they'll stay in the blood. We also have things like white blood cells, and white blood cells are sometimes even bigger than the red blood cells, and so they're going to also take a pass and they're going to continue on through the blood. Then we also have these other things called plasma proteins, and the larger plasma proteins are not going to be able to make it through any of those things. It won't be able to make it through the endothelial cells, the basement membrane, or the filtration slits. So what does end up making its way through, all the way through, it gets filtered. These things are going to get filtered through a little bit through the endothelial cells, a little bit more through the basement membrane, and then a little bit more through the filtration slits of the podocytes. And in the end, what ends up being filtered out, filtered into the glomerular space, so this is the glomerular capsule out here. What ends up in the glomerular capsule then is water, solutes, and ions. These things then are going to continue into the proximal convoluted tubule, where some of those things will be reabsorbed back into the body. Now we're going to take a look at filtration pressures. And the first thing I want to do is I want to compare the pressures in the glomerulus with pressures in other capillaries in your body. So there's the same factors in the glomerulus as there are in the other capillaries of your body. We have the hydrostatic pressure. Hydrostatic pressure is from fluid. And then we have a blood colloidal osmotic pressure, which opposes the hydrostatic pressure, and it's pressure due to materials in solution. So if we look at the two different types of capillaries we have, this could be our glomerular capillary, and then over here we can have our systemic capillary, which is your capillary that you find in your body, in most tissues of your body. And so the hydrostatic pressure is the pressure of the blood pushing against the wall of the capillaries and pushing fluid out. So that is what is called the hydrostatic pressure, or HP, hydrostatic pressure. Okay, and then the colloid osmotic pressure is due to the presence of these plasma proteins inside the capillaries. And what the plasma proteins do is they draw fluid back in because water always wants to move towards the solutes. Blood pressure in the systemic capillaries is fairly low. If you remember, we said at the arterial end it's 35 millimeters of mercury, and then as we get towards the venous end it ends up being 18 millimeters of mercury. And so it's fairly low blood pressure, and the reason for this is as the capillaries lead into the veins actually have low resistance. So there's not going to be a lot of blood that will back up into those systemic capillaries to increase the pressure. Blood tends to flow pretty easily into those veins. However, if we look at the glomerulus and we look at the afferent and the efferent arterial, and I just want to kind of draw this out here, we have the afferent arterial here. It has a bigger diameter, and it goes into the glomerulus, and then it goes out of the glomerulus through the efferent arterial. The afferent arterial has a much bigger diameter than the efferent arterial. So this is the efferent over here, right? And so all of this blood that's coming in through the afferent arterial into the glomerulus in here, it has to go out through that efferent arterial, and it's kind of like taking 3 lanes of highway into a 1-lane highway, and the blood sort of backs up into the glomerulus. And so the pressure in the glomerulus, those in the glomerular capillaries, is going to be higher just because of the diameter difference between the efferent arterial and the afferent arterial. So the hydrostatic pressure in the glomerular capillary will be higher, and typically the average hydrostatic pressure of the glomerulus, which we call the glomerular hydrostatic pressure, will be around 50 millimeters of mercury. So now I want to take a look at this picture, and I want to go over the other pressures that are involved in filtration. So we'll look at this picture and we'll kind of get our bearings first. This is the glomerular capsule. So inside the glomerular capsule, we have the capsular space. So that we can use as a big C, little s, meaning capsular space. Then we have the arterioles. So here's the afferent arterial bringing blood into the glomerulus. So I've really simplified the glomerular capillaries. Instead of drawing a whole bunch of them, I just drew one glomerular capillary so we can kind of see what happens. But there's a cluster of these capillaries, and all of these pressures will be occurring all over the glomerular capillaries. Then up here, I've drawn the filtration membrane. So you can see our podocyte here, you can see the basement membrane, and then you can see those endothelial cells as well there. And it's those 3 things that make up the filtration membrane, and that's what the fluid and the solutes and everything has to move through in order to get into the capsular space. So these are the pressures that occur. We have, first of all, we have the glomerular hydrostatic pressure. So that is the pressure from the blood. So the blood is pushing up against, it's coming in with every heartbeat, and it pushes up against those filtration membranes. And so I'm not showing the podocyte here, but you have all 3 of those layers that it has to get through. And then the amount of pressure that it takes to get through that membrane is called the glomerular hydrostatic pressure. And like I said before, the average glomerular hydrostatic pressure is 50 millimeters of mercury. That's how much pressure is pushing through that filtration membrane. Now, like I said, hydrostatic pressure is from fluid. So this fluid has higher pressure, 50, because the heart is beating and pushing that blood into that glomerular capillary. But out here in the capsular space, there's also fluid. And so this fluid is also going to exert a little bit of pressure. So that pressure we call the capsular space hydrostatic pressure. And that means fluid is pushing against the membrane here and will be pushing some fluid back into the glomerular capillary. So we have to take that into account. So we'll have to take the amount of fluid being pushed back in and subtract that from the amount of fluid that's being pushed out. And that would give us the net hydrostatic pressure, right? So let's go over that again. So there is a net hydrostatic pressure. If all we're looking at is hydrostatic pressure, the net hydrostatic pressure that is going to be the glomerular hydrostatic pressure from your heart beating and pushing the blood against the filtration membrane minus the capsular hydrostatic pressure, which is the fluid in the capsular space pushing back against that filtration membrane. That is your net hydrostatic pressure. So the capsular hydrostatic pressure is typically 15 millimeters of mercury. So our net hydrostatic pressure normally would be 50 minus 15, and that gives us 35 millimeters of mercury. So that is our average net hydrostatic pressure or net glomerular hydrostatic pressure. Now there is one other pressure though that we have to look at, and that is from those plasma membranes, or plasma proteins. So remember there's these little plasma proteins floating around in here, and they're acting as a solute, and so they're going to pull fluid back in. So we call that blood colloidal osmotic pressure, and it's the pull of water from the capsular space back into the glomerular capillaries as they're drawn back towards those plasma proteins. Now, typically, the blood colloidal osmotic pressure will be around 25 millimeters of mercury, right? So we have that. So now, if we look at this, we can see we have one pressure pushing fluid out into the capsular space, and we have two pressures pushing fluid or pulling fluid back into the capillary. So in order to get a net glomerular filtration, so net filtration, we have to find the net hydrostatic pressure and then subtract the blood colloidal osmotic pressure. All right, so we said the glomerular hydrostatic pressure was the blood pushing fluid out, and that's typically 50 millimeters of mercury. We said that the capsular hydrostatic pressure, the fluid in the capsular space pushing back against the filtration membrane, that pressure is 15 millimeters of mercury. And then we said the blood colloidal pressure, which is water being pulled back into that glomerular capillary, is 25, which would give us 35 minus 25, which gives us 10 millimeters of mercury. So 10 millimeters of mercury is our net filtration pressure at the glomerulus. So if we want to compare the glomerular pressure with the systemic capillary pressure, we would say that here's the glomerulus and the pressure is 10 millimeters of mercury. Going through what we just went through, talking about the glomerular hydrostatic pressure, taking into account the capsular hydrostatic pressure, and also taking into account the blood colloidal osmotic pressure. So with all those things, we end up with 10 millimeters of mercury. If we look at the systemic capillary, what we found was that the capillary hydrostatic pressure at the arterial end was 35, and then the blood colloidal osmotic pressure was 25. If we looked at the venous end, we saw that the hydrostatic pressure in the capillary decreased to 18, and the blood colloidal osmotic pressure stayed the same at 25. So if we wanted to look at all of the hydrostatic pressure in the capillary, we take 35 minus 25 and add that to 18 minus 25, which gives us 10 plus a negative 7, which means our net pressure in our systemic capillaries is 3 millimeters of mercury. So I'm just comparing these two. So this is your systemic capillary, and then this is your glomerular capillary. So the point being that the pressure in the glomerular capillary is much greater than what we see in your systemic capillaries. And that's why we end up with what we call filtration. And this only occurs then at the glomerulus, in that renal corpuscle across that filtration membrane. So 10 millimeters of mercury is the average pressure that's forcing water and solutes out. Out of the blood and into the capsular space. So if you have a problem with the filtration pressure, then that's going to affect your kidney function. So the glomeruli, all those little glomerular capillaries, they generate 48 gallons of filtrate a day. That's a lot of. So this is saying what the glomerular filtration rate is. So per day, the filtration rate would be 40 gallons of filtrate is filtered per day. But we normally don't look at the glomerular filtration rate as how many gallons per day. Instead, we look at it as how many milliliters per minute. The glomerular filtration rate is equal to the total of the filtration rates of all of the functioning nephrons in the kidney, and it's really considered the optimal way to measure kidney function. The glomerular filtration rate can be measured usually by using an exogenous filtration marker like creatinine. Creatinine is a substance that is almost 100% excreted and not reabsorbed. So you can measure how much creatinine is in the blood and then measure how much creatinine ends up in the urine, and you can figure out then what the clearance rate is, or the glomerular filtration rate is, of the kidneys. For example, if you have a person who eliminates 84 milligrams of creatinine per hour, and that person has a plasma concentration of 1.4 milligrams per deciliter of creatinine, then that will give you how many deciliters per hour of creatinine is being cleared by the kidneys. And 1 deciliter per hour is equal to 1.67 milliliters per minute. So then you can look at the GFR and calculate that in mils per minute, which in this case then 60 deciliters per hour would be equal to around 100 milliliters per minute. You get this by multiplying the 60 deciliters per hour times the 1.67 mils per minute so that you can come up with the mils per minute. So in this case, you multiply 60 by 1.67 and you end up with 100.2 then mils per minute. So a normal glomerular filtration rate is going to vary according to age and sex and body size. In young adults, it's approximately 120 mils per minute, and it will decline in normal individuals with age. But a decrease in the glomerular filtration rate might be a marker of kidney disease, and usually it precedes the onset of most kidney failures. So it's a really important thing to look at the GFR. Clinically, a patient will be told if their GFR is between 60 and 120, then that would be normal. If it's between 15 and 60, then that is kidney disease. And if it's between 0 and 15, that means that there is kidney failure. So going back to our 10 millimeters of mercury is the average glomerular filtration pressure. Okay, so not rate, but pressure. Anything that affects that pressure is going to affect the glomerular filtration rate. So any factors that alter the net filtration pressure will alter the glomerular filtration rate. So let's look at a couple of examples here. Let's look at one example at least. So here you have your capillary, your glomerular capillary, okay, and then here we have the capsular space, and that's leading into the proximal convoluted tubule. And so what we said was the glomerular hydrostatic pressure was 50 normally, so that's the glomerular hydrostatic pressure. We said that the capsular hydrostatic pressure is normally 15, and we said that the blood colloidal osmotic pressure is typically 25. And then we said, well, if we take the net hydrostatic pressure, which is 50 minus 15, and we subtract the 25, we end up with 10. So let's take a situation where the blood pressure drops. So say your blood pressure decreases by 20%. So that's going to change the glomerular hydrostatic pressure because that's caused from heart pressure, right? That's caused from the blood pressure, the pressure of the heart beating and pushing that blood into that glomerular capillary. So if we take 20% of 50, 20% of 50 is 10 millimeters of mercury, and so we're saying if our glomerular hydrostatic pressure drops by 10 millimeters of mercury, which would be, 20% of it, then we're down to 40 millimeters of mercury. So if our pressure here, if our glomerular hydrostatic pressure drops down to 40 millimeters of mercury because your blood pressure decreased by 20%, now we take a look at that same equation and we have 40 minus 15 and then minus 25 because nothing has changed there. And now we have a glomerular filtration pressure of 0. So at this point, then kidney function stops, right? Your kidney function stops. And so now this can happen, blood pressure decreasing by 20%. It's not a normal thing that happens, but it can happen because of things like hemorrhaging, losing a lot of blood. Your blood pressure is going to drop. Shock, which is blood pressure dropping very rapidly. Dehydration, where you just don't have enough fluid to give you enough blood pressure. All of those things can cause a dangerous decrease in the glomerular filtration rate because they are affecting the glomerular filtration pressure. And so it can lead to, all of these things then can lead to acute renal failure as the kidneys fail. So now we have to look at the regulation of the glomerular filtration rate, right? So we're going to look at regulation of the rate. So there are 3 levels of control that will affect the glomerular filtration rate. The first one is called autoregulation. And autoregulation includes things like the mesangial cells. So I'm going to show you what those are. And also the afferent arteriole. So we'll look at how these 2 things can help to auto-regulate the glomerular filtration rate. And then if autoregulation is ineffective, then a wider response is going to be necessary. And so then we're going to look at the hormonal regulation. And the hormonal regulation of the glomerular filtration rate includes 2 things. It includes the RAAS, which is the renin-angiotensin-aldosterone system, and it also includes the natriuretic peptides from the heart. And then in addition to this hormonal regulation, there's also a neural regulation. The neural regulation is really just the sympathetic nervous system. So we're going to look at how the sympathetic nervous system regulates the glomerular filtration rate as well. So let's take a look at this picture again, and I want to go over a couple of other things that are on here. So again, this is our glomerular capillary right here. This whole thing is. And I'm just showing one capillary, although we know there's a bundle of capillaries. And then there are these cells, and I drew one here. So there's the cell, and then there's the nucleus of the cell, and that cell is called a mesangial cell. And this mesangial cell will contract, and when it contracts, it will narrow the diameter of the glomerulus, of that glomerular capillary. So when the capillary diameter decreases, then the glomerular hydrostatic pressure goes up. So when the mesangial cell contracts, so it would be contracting, and it's going to be narrowing then the passageway, the lumen of the glomerular capillary. And just by narrowing that lumen of the capillary, now the blood has less room to bump around in, and so your glomerular hydrostatic pressure is going to go up. Okay, so mesangial cell contracting increases glomerular hydrostatic pressure, which will increase the filtration rate. So these are autoregulation. So one way to increase the glomerular filtration rate is for the mesangial cells to contract. Right? The second way is that, and this is going to occur if your blood pressure drops. So if your blood pressure decreases, then the mesangial cells will contract. It will narrow the lumen and increase the pressure inside that glomerular capillary, and then your hydrostatic pressure, your glomerular hydrostatic pressure will go up, which will then increase your glomerular filtration rate, how fast that fluid is being filtered. So then if you have the other situation where if your blood pressure increases, so now you have increased blood pressure, the smooth muscles lining the afferent arteriole will contract, and when they contract, they will narrow the lumen of the afferent arteriole. So if the lumen of the afferent arteriole decreases, and we go from this wide to only this wide, now there won't be as much blood that can get in through the afferent arteriole to get into those glomerular capillaries. So in that case, just by by the afferent arterial smooth muscles contracting, it will decrease the blood pressure. So again, if the blood pressure increases, the afferent arterial will vasoconstrict. That narrows the lumen of that arterial, less blood can come in, and if we have less blood coming in, the glomerular hydrostatic pressure will go down, and if the glomerular hydrostatic pressure goes down, that means that the glomerular filtration rate goes down. Not as much blood means not as much being filtered. Now, if autoregulation is ineffective, meaning if the mesangial cells can't fix the problem when blood pressure goes down, and if the autoregulation of the afferent arterioles doesn't fix the problem when blood pressure goes up, then something else has to happen. So if autoregulation is ineffective, then a wider response is necessary. And so the wider response can be either hormonal or neural. So let's take a look at hormonal. So the hormonal response to regulate the GFR is either by using the RAAS system, which is renin, angiotensin, aldosterone system, or it is using the natriuretic peptides. The RAAS system is more of a long-term solution. But we also have a neural solution as well. So neural response would be by the sympathetic nervous system. By activating the sympathetic nervous system, it will also affect the glomerular filtration rate. So the RAAS. So this system begins with the hormone called renin. Renin is a hormone that is released from the kidney under several different situations. It will be released when the blood pressure decreases. So if your blood pressure decreases, it goes down, then renin gets released. If you have a decrease in blood volume, renin will be released. If you have a blockage of the renal artery, then there's not as much blood getting to the glomerulus, then renin will be released. Or if there's a blockage in one of the branches as well. So that's one way, that's one reason why renin would be released from the kidney. All of these things are bringing less blood into the glomerulus. So the kidney is thinking that it needs more fluid coming in, and so renin will be released. Another thing that will cause the release of renin is a stimulation of the juxtaglomerular cells by sympathetic innervation. So if the sympathetic nervous system is activated or is stimulated, your fight or flight, then renin will be released from the kidney as well. The sympathetic nervous system, if you remember, when it is stimulated, it causes vasoconstriction. So there will be vasoconstriction of the afferent arteriole. That means that less blood is going into the glomerulus, and that's going to cause the kidney to release renin. And then lastly, the last reason why renin would be released is if there was a decrease in the osmotic pressure in the distal convoluted tubule. So in this case, the macula densa cells detect that there's not as many solutes in the distal convoluted tubule. And in order to bring the concentration of solutes back to normal, then renin will be released so that more fluid will be reabsorbed, and that will bring that solute level concentration back to where it should be to bring the osmotic pressure back up to where it should be. A lot of times these factors will all be interrelated, meaning one might cause the other to happen. But the end result is always going to be the same, and that means that renin will be released. All right, let's take a look at the RAAS. Renin, angiotensin, aldosterone system. So this system responds to decreased blood pressure. Talked about that decreased blood pressure, decreased blood volume, blockage of the renal artery. So we talked about those things that will trigger renin to be released. So when you have decreased blood pressure, that means you also have decreased blood flow to the kidneys. If you have decreased blood flow to the kidneys, this means there will be a decreased glomerular hydrostatic pressure, which leads to a decreased glomerular filtration rate. So the whole purpose of the RAAS system then is going to be to increase the glomerular hydrostatic pressure and increase the glomerular filtration rate. So let's look and see how this happens. So the first thing is when you have a decrease in blood pressure and there's not as much blood coming into the kidneys will release renin. So remember, renin is released from the juxtaglomerular cells. Right? And so renin gets released into the blood. Now in the blood there are these plasma proteins that are called angiotensinogen. This is a plasma protein. And renin causes angiotensinogen to be converted to angiotensin 1. Then angiotensin 1 travels through the blood to the lungs. And in the lungs, angiotensin 1 becomes converted to angiotensin 2. This happens by an enzyme which is called ACE. ACE stands for angiotensin converting enzyme. Angiotensin 2 is a major peripheral vasoconstrictor. It also constricts the efferent arteriole. So if we take a look at this and we look at, here's your blood vessels in your peripheral system, and then they become vasoconstricted, so they become much smaller. So this means that these blood vessels, which are going to areas like your skin and your muscles and areas like that, they're not going to receive as much blood because these blood vessels are constricting. But what it does do then is it pushes the blood towards those blood vessels that are not vasoconstricted, like the renal artery. So the renal artery is not vasoconstricted, so we get increased blood flow to the kidney. At the same time that more blood is going into the nephron, then we also have the efferent arteriole, which is vasoconstricted. So that's causing increased resistance. So the blood, we have more blood coming in, and it's making it harder for blood to go out. So the blood gets trapped in the glomerulus. That will increase that glomerular hydrostatic pressure, which will increase the glomerular filtration rate. So we went from having decreased blood pressure, decreased blood flow to the kidneys, and a decreased glomerular filtration rate, and through the RAAS system, we end up with increased blood flow to the kidneys, increased glomerular filtration rate. So this system is in place when your blood pressure drops to make sure that the kidneys have enough hydrostatic pressure to have a normal glomerular filtration rate. So to kind of do a recap of this, if we look at the body's response to decreased blood pressure, that's when the RAAS system kicks in. And that's because if you have decreased blood pressure, that means you have decreased blood flow to the kidneys. So we get this endocrine response with the RAAS. And that's where renin stimulates through that process of activating angiotensin II. And that leads to an increase in blood flow to the kidneys. But there's something else that renin does as well. Renin also affects the central nervous system. Renin targets the thirst centers in the hypothalamus, and it makes you thirsty. So if you're thirsty, you're going to drink more, and that will increase your fluid consumption. If you bring in more fluids, that's going to help to increase your blood pressure, your blood volume, which will then increase your blood flow to your kidneys. In addition to that, renin also causes the release of ADH. So ADH gets released from the posterior pituitary. ADH causes increased water reabsorption in the kidney. If you're increasing the water reabsorption, that means you are increasing the amount of blood volume. That means you are increasing your blood pressure. Then another response of the nervous system is that the sympathetic nervous system is activated. The sympathetic nervous system will increase cardiac output, and quite a bit of the cardiac output goes to the kidneys. So you're going to increase the amount of blood flow to the kidneys. And that will increase the glomerular filtration rate. So now if we look at the opposite, if you have an increased blood pressure, then the natriuretic peptides will come into play. So these natriuretic peptides are hormones that are released from the heart. The heart has to pump the blood through it, and if the right amount of blood is coming into the heart can beat normally, it can pump the blood normally, but if too much blood comes into the heart, now it's going to have decreased function. It's not going to be able to beat as effectively as it should. So the natriuretic peptides will be released from the heart in response to increased blood pressure. So you have too much blood pressure and the walls of the heart are saying, there's too much blood in here and I can't beat properly. So something has to be done about this. So the atrium and the ventricles will release these natriuretic peptides. The atria release a natriuretic peptide called ANP. This stands for atrial natriuretic peptide. And the ventricles will release a natriuretic peptide called BNP, which stands for brain natriuretic peptide. So let's talk about them separately. So when the atria release ANP, ANP does two things. It dilates the afferent arteriole and it constricts the efferent arteriole. So again, we are having the same situation here where the afferent arteriole becomes dilated, so we have more blood flow going into the glomerulus, and the efferent arteriole becomes constricted, so there's more resistance, and that is going to increase the glomerular filtration rate. So there will be more water flowing into the tubule, which means there will be more water lost in the urine. And when you lose water, then your blood pressure will go down. So we end up with a decreased blood pressure. Now the ventricles, they release BNP. So when BNP gets released, it does the same thing, but it does it in a different way. BNP will decrease sodium reabsorption at the renal tubules. So if in the tubules sodium is not being reabsorbed and sodium ends up going in the urine, water is going to follow along with the sodium, so we get an increased urine output because we get increased water being lost. And whenever you lose water, again, you decrease blood pressure. So when you lose water in the urine, you're decreasing blood pressure and you're decreasing blood volume. In both of these cases then, you started out with an increased blood pressure, and through the ANP and the BNP, you decrease the blood pressure and you decrease the blood volume. Now let's talk about neural regulation. So neural regulation is going to be autonomic regulation because it involves the autonomic nervous system. And so the autonomic nervous system will regulate the GFR. So most of the autonomic nervous system or innervation of the kidneys is the sympathetic nervous system. Sympathetic activation causes powerful vasoconstriction. Of the afferent arteriole. So we have normally where the afferent arteriole is bringing a proper amount of blood into the glomerulus. Now we have a powerful vasoconstriction of that afferent arteriole, and that means that there won't be as much blood going into the glomerulus. This sympathetic activation, it overrides any local regulation. Because it is vasoconstricting that afferent arteriole, that means not as much blood is coming into the glomerulus, and that means that the glomerular filtration rate will decrease. This happens, for example, in an acute decrease in blood pressure. So if your blood pressure really drops drastically, or if there's decreased cardiac output because of a heart attack. So the sympathetic nervous system, which is your fight or flight nervous system, is going to want the kidney to function less. It's not so worried about the kidney producing urine at this point. It's more concerned with areas like your brain getting enough blood and oxygen, and the heart getting enough blood and oxygen. And so it's going to override any local regulation to make sure that the blood is being diverted to organs other than the kidney. As the crisis passes, then the sympathetic tone will decrease, meaning the sympathetic nervous system starts to decrease in function, and the filtration rate will then return to normal. There are other times that the sympathetic nervous system also alters patterns of blood flow, which can affect the blood flow to the kidneys. For example, with the sympathetic nervous system, and I'm talking about things other than a crisis, but for example, if there's warm weather, in warm weather, the superficial blood vessels dilate, so blood gets directed to the skin and away from the kidney. So for a short period of time, just a temporary period of time, the glomerular filtration rate will decrease. Another example would be during strenuous exercise. So during strenuous exercise, again, blood is going to be diverted away from the kidney. It'll be shunted away from the kidney, and it's going to be diverted to the skin. So that you can regulate your blood temperature. And so again, it's going to decrease the glomerular filtration rate temporarily. And then as soon as you are resting again, then the blood flow will be directed back to the kidneys again. At maximal exertion, the renal blood flow could decrease by to 25% of what it is normally. So this can be a problem for endurance athletes. So in endurance athletes, of course, they're going to have this decreased glomerular filtration rate, but because they're endurance athletes, that means that they're going for longer periods of time. And what that means then is if you're decreasing the glomerular filtration rate, that means that metabolic wastes are going to build up in the body. And that also means that protein might occur in the urine. We call that protein urea, when protein shows up in the urine. This can happen because when there's low oxygen, which we call hypoxia, that can damage the glomerular cells, and then protein can sneak through that filtration membrane. So the other thing is that if there's enough damage, then blood will show up in the urine, and that is called hematuria. And hematuria develops in about 18% of marathon runners. Most of that, they believe, though, is from trauma to the bladder, because running is a high impact exercise, and so the bladder receives a lot of trauma, and so blood might be released from the lining of the bladder. But that's more likely, but there is a small number of athletes and runners who can experience acute renal failure. And again, this is because for too long a period of time, they have had a decreased glomerular filtration rate, and that will damage those cells of the kidney. That's gonna damage the glomerular cells, and then red blood cells are going to leak across that filtration membrane into the urine. Okay, then that ends this video for the urinary system. And then in the next video, it's probably going to be a little bit shorter than this one, but we're going to go ahead and look at the rest of the tubule and look at reabsorption and secretion. So we'll see you next time.